it was a very kind of uncertain time for me. But I was fortunate. I think the only reason I was able to get through that time period is because of Bob and other people in the department who were supportive of me when maybe I was really scared and uncertain about that.
Hello and welcome to another episode of The Next Step. I'm Jeff Kozlowski, your host. I'm excited to welcome our next guest, Makaia Papasergi-Scott, Assistant Professor of Biochem and Biophysics, as well as Pharmacology and Physiology here at the University of Rochester Medical Center. First, I also want to recognize that we have our first co-host on The Next Step, our first faculty co-host, Bob Freeman, who is a professor of pharmacology physiology. Thank you so much, Bob, for helping me out today.
My pleasure.
And can you first just sort of tell us a little bit about your work here at University of Rochester, what brought you here,
Sure.
all that good stuff?
All right. Well, I'll keep it very short. I came to Rochester just about 31 years ago now, and I came after a postdoc at Washington University in St. Louis. I had been doing research on neurodegeneration, specifically cell death in specific parts of the nervous system, and trying to understand those molecular mechanisms. So I started my lab here, started my research here back in 1994 in the Pharmacology and Physiology department. And of course, one of our most productive and really stellar graduate students was Makaia many years later. About 8 or 9 years ago, I was winding down my research. Makaia actually joined my lab for a short stint, and we'll be discussing that. And I focus much more on teaching. So I spent a lot of time in this particular room, actually, over the last 7 or 8 years, teaching medical students pharmacology. Recently, became a part-time faculty member in the department, and am spending a lot of time outdoors on the ski slopes in particular this time of year.
All right. Well, thank you again for
Sure.
taking some time off the slopes and joining us. Makaia, let's get started with your story. I think a good place to start is the fact that I know you had a very sort of set plan for your career, like before coming to U of R, right? You know, you were going to go to college, get a PhD, do research, with an ultimate goal of curing neurodegenerative diseases, right? Can you talk through just kind of how that came about, how that sort of vision formed and what that looked like exactly?
Yeah, absolutely. So it started pretty early. I think I was in 8th grade in grade school. And I grew up in a kind of a small town. I don't even think I was that good of a student in 8th grade, actually, until I got to 8th grade science class, which was taught by Mr. Chris Moore. And he really inspired me to like science. And also inspired me to be a better student, which was perfect timing because it's before I got into high school and when all of your grades actually start mattering. That was like kind of a first initial push. And then when I got into 9th grade, there were kind of like two parallel things that were happening. I was getting more and more into the science and really enjoying the science classes. But also we were assigned a project in English class. And so in that English class, it was, who do you think is a hero? And write kind of like a news article about this person that you think is a hero. Who is your hero? And so I picked Michael J. Fox for that. I think I really liked Back to the Future, and that might have started that, thinking about that, but I found out he had Parkinson's disease. And kind of learning about his plight with Parkinson's disease, that when he found out that he had Parkinson's, he didn't kind of take that as, this is the end, I'm just gonna sit here and accept it. He actually started a foundation, and really was like, you know what, I'm not going to just sit here, I'm going to actually think about cures. And so I was really inspired by that. And then as I progressed through high school and took chemistry and biology courses, I really realized that neurodegenerative diseases, I was really interested in that. And I was like, you know what, I can do something. Like the same way that Michael J. Fox was thinking about, how can we do something? I myself am at the stage of my life where I could pick what I want to do with my career and do something about this. And so that really kind of set me on a trajectory for wanting to do that. And it became kind of like this vision and a goal that I had for my life. that I was like, someday I'm going to cure neurodegenerative diseases, not just one. I was very ambitious as, a high schooler, thinking like, I'll just cure all of the neurodegenerative diseases. I'll win a Nobel Prize for it. And that's what I'm going to do. And so every decision I made kind of stemmed from that. It's like, how do I get to this goal? And so I ended up going to Geneseo, about half an hour south, SUNY Geneseo, majoring in biochemistry. And I had found out that a professor there was studying Alzheimer's disease, amyloid beta fibrillogenesis, and the reversibility of that. And so as soon as I got there, I went and met with this professor to just talk about his research, Dr. Kazu Shiji Yogayama, and he looked at me and he's like, okay, you start next semester. And so I ended up doing research for 3.5 years on that project, and it really solidified that I want to be in research and be doing this. And so that's really where I started. It's kind of like, a trajectory of having some really great mentors and things happening in my life, really.
Okay, first of all, Back to the Future. 1, 2, or 3 is the best. What's your favorite?
The first one's probably my favorite,
Yeah.
but I like all of them. I think it's a fantastic trilogy in general.
They're all great. They're all great. I know I kind of lean toward 2, but they're all great. Okay, second. So then you get to graduate school here at School of Medicine and Dentistry. Requested interviews with all faculty doing neuroscience research. Come to find out of the 4 interviewers, only 1 actually did that and was not taking students. So what was it like finding that out? You might now have to veer from the path a little bit.
So I was a little bit gutted, I think, at first. Actually, Bob was the only interviewer that was working on neuroscience work. And that year he had decided he wasn't going to be taking students. And I had other interviews at other places where I could have gone into research doing exactly what I wanted to research. I could have studied Alzheimer's and Parkinson's disease. There was something about the University of Rochester that I just felt like it was home. So I kind of had to rationalize with myself, well, I'm not necessarily going to be able to study exactly, the thing I thought I would be studying, but this is a training period. I'm going to get the tools, and then after graduate school, I'm going to go back to doing the thing, still working towards the path, but right now I'm developing the tools that I need in order to do that. And so I kind of rationalized it to myself. It's like, this is just a detour in a way, but we'll get back to that. And this is really, still progressing towards that ultimate goal.
You mentioned University of Rochester feeling like home. Anything in particular make it stand out like that?
I think it was the people. I just felt like I fit among the students and also the faculty. I thought the faculty were really approachable that I had talked to during interview weekend. And I felt that the students, they had the right kind of similar mix that I had. They had lives, but they really were very serious about studying and doing research and what they wanted to do with their lives. And so I felt that resonated with me more so than other places that I had gone to.
Well, ultimately, then you joined a lab of a gentleman by the name of Dr. Greg Tull, who eventually relocated, and we'll talk about that in a little bit. So that shifted your focus to G-protein coupled receptors. Not what you initially envisioned doing, but captured your attention somehow or a little bit. So how did you, stay engaged in those early years? So
that was actually probably my first exposure to starting to see science and another type of research that I was really excited about that wasn't this neuroscience and neurodegenerative research. The first time I had met with Greg, which actually he was one of my interviewers, we had talked about the research he was doing in his lab, and I remember just being really enthralled by just the type of research he was doing. The work that was being done in the lab and the work that I did during my thesis work was on this protein called RIC8, which is a chaperone that folds G protein alpha subunits. And so the reason this protein is important is because it's what acts inside the cell after G protein coupled receptor is activated. And G protein coupled receptors are targeted by 30% of FDA-approved pharmaceuticals. They're in pretty much any part of your body in physiology, And pretty much any disease you can think of has a GPCR either involved in its pathology or in the treatment of that disease. And so, again, I was doing kind of this rationalization. I was like, you know what? GPCRs are involved in neurodegenerative diseases or the treatment of it. So I'm not really veering away from my ultimate goal. I'm really kind of staying on the same track. It just doesn't look as straightforward. You know, this whole time I'm kind of rationalizing this in my head. And I also felt that his lab, the group of people he had in the lab, really felt like family to me. We really fit together and had fun together and enjoyed each other's company and kind of had the same mindset. I think it was a very hardworking lab, but also had a lot of fun. And that was definitely appealing to me, and I could see that was a good training environment.
And eventually Dr. Tall relocated to Michigan, and you had the opportunity to join him. Ultimately decided to stay back here in Rochester. What went into that decision to to stay back here and finish here?
It was a really difficult decision. I think it was motivated a lot by things that I was going through personally during that time, though. So I had just gotten married, actually, about, I think, a month before he decided to move. And my husband had a full-time job here. And also, I had a death in my family around that same time, like within a week of me getting married, actually. And so there's this kind of nagging feeling, how many lives am I going to miss out on for pursuing this dream and this research and wanting to do this? And so I was really struggling, actually, with, is it worth it, what I want to do? And so suddenly staying close to the family became much more important. And since I grew up in Western New York, I was relatively close to the family here. And so those were things that were weighing on my mind as I was making that decision. And I didn't quite think, I think, when I made the decision that I would feel displaced. That certainly came as, you know, I got closer to, the lab was moving and I wasn't going with them. But I kind of had already made my decision at that point.
Were you the only one that stayed back?
Yes.
Yeah.
So we had one graduate student who graduated a little bit, you know, rushed to get out the door, and then another student who actually had moved with Greg to Michigan.
So, you know, once you made the decision, though, and the lab had officially relocated, what was it like kind of, you know, navigating the months and whatnot after that?
It was really difficult. I remember the first day that the lab had actually moved, and I went and I was adopted. I affectionately refer to Bob as my adoptive graduate advisor. And I was essentially in his space then doing research in his lab. He was my co-advisor. And I remember the first day that like the lab had actually moved. My old lab was empty, and all my stuff had been moved to Bob's lab. And I sat down at the desk and just felt so alone. Like the people that I thought were my family were suddenly gone. But I will say that Bob actually had his last graduate student before me had just finished. And I think she had moved like within a couple weeks of me joining before I joined the lab. And she had actually written me such a nice little note. Gave me a little card with a keychain. And she wrote, you know, I know you didn't start here, but you're going to be using the space, and I want you to know that you're part of this family. And she put the entire line of Bob's graduate students and included me and said, this keychain has been passed down through all of Bob's graduate students, and it's now yours to give you luck. And if he has another graduate student after you, then you're expected to pass it on to them. And that part was like, I think that also defines the welcoming environment that I had, not just in Bob's lab, but the university and the whole department, pharmacology department in general. There was a number of labs who let me use their equipment and things like that, which helped a little bit, but I still had this, you know, it was an internal struggle that I had. And I don't think a lot of people saw that at the time.
Bob, before you jump in, can you talk a little bit about, like, from your point of view then, what was it like? Did she come to you and was like, here's my situation, can you help? Or, you know, what did that time look like?
No, it was very much a conversation, and you know, I could tell that Makaia was very, or felt that she would be comfortable in that environment. She was certainly going to have a lot of space and a lot of time to herself, because my lab was pretty much winding down at that point. And so whether that was a good thing or a bad thing, I'm not sure. I've always had a very open door, just walk in when something's not working, walk in when something's working, when something's going on that has nothing to do with work, but it's, taking your mind off of your research. I think that is appreciated, I hope. But it was a kind of a logical place. My background was molecular biology, but also biochemistry. Makaia was very much trained as a biochemist in our department. So many of the tools, many of the sort of things we think about, not just the physical equipment and reagents, but what kind of our mindset in terms of thinking about scientific problems. I think we had a lot of parallels there. So it was easy to converse. And I think over time, I think you began to feel pretty comfortable, hopefully, at least coming to me. And certainly I wasn't the sole person that you utilized as a resource after the Tall Lab moved. Can you share a little bit about some of the people in the department that you really relied on, even more so when you were working on your own?
Yeah, so Bob Dirksen was definitely very supportive. He was the chair at the time, and so he would kind of check in with me and see how I was doing. It was always, I think he's always been kind of a fan. It almost felt like he was like supporting me and like a fan of, helping me. Probably all the students say that, I don't know. But I feel that way. I felt that way. And I feel that, there was definitely resources. Paul Brooks actually had a piece of equipment that he said I could continue using. We had used it when I was a graduate student in Greg's lab, and he's like, you can continue using that. And a number of other labs that microscopy, there were people who stepped up and let me use their microscopes and things like that. I had developed good relationships with my peers that weren't working in the same lab as I was. And so those relationships continued, and they were also really supportive of the situation I was in and making me feel like I still had a place. It was still initial kind of like, you form very close bonds, I think, with your lab when you're working there for a number of years. And the being able to run down the hall initially, and talk to your graduate advisor about whatever it was. And I think I eventually started doing that not long after I think I got there into his lab. But at first it was difficult. It was like you have to learn kind of a new environment and kind of a new like relationship and how you're going to navigate that. And thankfully Bob is very, very good at that. And I was fortunate to be in that situation. Especially when I was struggling with some really dark things, feeling displaced, but also personally, I was still going through all of that and struggling with, thinking about my career and thinking about what am I going to do and what does this mean for me in general. It was a very kind of uncertain time for me. But I was fortunate. I think the only reason I was able to get through that time period is because of Bob and other people in the department who were supportive of me when maybe I was really scared and uncertain about that.
Shout out Bob Dirksen real quick, because I feel like his name has come up in that capacity several times in this podcast's life. So Bob, he's doing something right out there.
Yeah, he certainly is. He is very much, if you are putting out effort and are contributing and sort of making the environment a great place, he will go way out of his way to support you and make sure that he can do whatever he can to make you as successful as possible. And it goes right down to certainly to the graduate student level, but to our technicians, to the staff, to the faculty. So great chair. So Makaia, when you were finishing your PhD, of course, my main role was not so much as a day-to-day advisor on the science and the project you were working on, but more so, I think, in a lot of ways, to have someone to hear you or as a sounding board and to reflect on a little bit about some of the difficulties, but also where you want to go in the future and what's your path going to look like and what are the next steps. Can you just talk to us a little bit about that evolution, your thoughts about at that time, what are the next steps and how am I going to get there?
Yeah, absolutely. So as I was going through this kind of struggle and this period of kind of like darkness in my personal life and thinking about, is this career that takes so much out of me? I didn't go home when that family member who had passed away, that preceding year. I was so busy with work and my graduate studies, I didn't go home.
Mm-hmm.
And that really affected me. Kind of changed my outlook a little bit. Like, is this worth it? And so as I'm thinking about those things, and I'm also seeing kind of the struggles that academic faculty go through at an R1 institution, that these research-driven institutions, faculty members are essentially wearing every hat possible, and one of those hats is, needing to get funding and always worrying about funding your lab, and if you don't get funding for your lab, you have to let people go who maybe don't deserve to be let go and things like that. And I'm seeing some of these things happen, and I'm starting to think, is this really what I want? And then I was also thinking about it, and I was like, okay, well, what would also be still working kind of towards this pathway? Again, rationalizing
Yes.
things in my mind and kind of doing some acrobatics. Well, I could work at more of an undergraduate university, have a primary focus of teaching, because I really enjoy, kind of that teaching aspect of academia. I liked the idea, and still do, of being able to inspire the next generation. I thought, well, what if I'm not the one that cures these diseases and is the one who's going to get the Nobel Prize, but what if I'm able to inspire the next generation of scientists? And I thought, well, wouldn't I be more impactful if I get to do that, lecture to 200 students and put them all out there towards this goal? And so I'm still rationalizing and kind of doing some of these acrobatics and thinking yes, I can change my career focus a little bit. I'll still, at a lot of these undergraduate institutions, like where I came from at SUNY Geneseo, people have these research labs. So I could still do research a little bit and then do the teaching and inspire students. Thankfully, too, Bob was super supportive he's an excellent teacher and has taught many courses. And so I was able to TA one of his classes. And he was very supportive of if there was opportunities for teaching and getting me in touch, too, with some of his former students. And other students in the department that had gone into teaching after being graduate students here. And so I got to interview them and see what it was like for them to do that. And so at the time I was like, oh, this seems like, maybe that's what I want to do with my career. And so I started reorienting, okay, now how do I get to this new, still on the same path, it's kind of the same goal, but I'm going to do it slightly differently. And so that's really how that evolved, I suppose, during that time.
And I think it's worth noting that while you were exploring these alternative career opportunities, really teaching opportunities, and had several that were not at our university but neighboring institutions, you didn't drop the ball at all in the lab. She was pretty much in the lab as much as ever, and working just as hard, and then would run off at whatever time, 6 o'clock at night, to drive to a nearby community college to teach a course.
That is a grind.
Yeah.
And so you eventually did get some adjunct teaching roles, started a first postdoc role here at URMC. Talk about, what you learned during that experience and just kind of what sort of came out of that new path.
Yeah, and so then I was looking for as well, I was like, what, I still should probably do a postdoc before I go on to getting a full-time teaching position. And I still need more teaching experience. And so Hugh Schott, who is also in the pharmacology department, he worked down the hall actually from Bob's lab. His office was down the hall from Bob's lab. And he was a new faculty member at the time, a fairly new faculty member at the time. And he worked on neurodegenerative diseases. So, of course, I could get back on track with the research focus that I had intended to do since I was in high school. It was an interesting project for research, and he also was very supportive. He understood my career goals at that time, that I wanted to go into teaching, and so he was okay with me being an adjunct, teacher at a couple colleges locally. And so I was able to get experience at Genesee Community College and SUNY Geneseo. And some of my days were kind of crazy. In the morning, I would be at Geneseo teaching a lecture And then I would come back, do some lab work, and then in the evening I was teaching a night course at Genesee Community College, and I don't recommend that to anyone.
You probably didn't see your husband much at that time.
No, I don't think I did. But after grad school, he might have gotten used to that.
There you go.
So, I was kind of juggling all of these things. And one of the big components of research in that lab was actually doing mouse work. And that was something that I hadn't experienced before. I hadn't worked with mice. And I realized fairly quickly that actually that was not something that I wanted to do in my research. I don't have anything necessarily against research using mice and rats and things like that. But for me, it bothered me to be able to have to work with mice and rats and dissect out the brain, and it wasn't for me. And so I was working through that, and I was kind of missing the research that I did in graduate school, and I was a little bit frustrated, I think, and I was enjoying the teaching. I really do enjoy lecturing. I noticed too that in that lab and also in my lecturing, the interactions that I really liked were the one-on-one interactions, either when the students came to me during office hours in the courses I was teaching, or in the lab, I had students that were younger students that I was given to kind of train, and I really enjoyed that. I was confronted one day too, Sandeep, who is in our department, has been a staff scientist in our department for a long time. He looked at me one day and he's like, "What is this face you're making? Are you sad, angry, frustrated?" And I was like, "I'm all of those things." I'm all of those things. And so I really started to reflect on, why am I feeling this way? I'm doing the thing that I thought I wanted to do. I feel like this is I thought I knew what I wanted to do now, kind of career path. And I was also reading at the time this book called Women Don't Ask. I had gone to a GWIZ, Graduate Workers in Science seminar. And there's this woman who talked about how that had really inspired her, and that was
the book,
Women Don't Ask, to start negotiating for herself. And this isn't going to negotiating for myself, but in that book, the very first part of it says, before you negotiate, you need to know what's going to make you happy. And so I got to that part of the book, and I just thought to myself, I was like, do I even know what would make me happy right now? And so it was really a period that I was reflecting, and I thought, I miss the research I was doing before. I can't kill mice. And I started to come to the terms with the fact that I don't know if I'm a neuroscientist or that I even want to be a neuroscientist. Okay. And it was actually a little bit of a period of mourning, I think, for me, to let go of that previous dream that I had had and to accept that, I miss this former research I was doing. I miss G proteins. I miss G protein coupled receptors. And the type of mentoring and the teaching that I like the most is that one-on-one interaction. It was like, well, that sounds like what my graduate advisors were doing. And so it was kind of like this sudden realization, you know what, I think I need to do a second postdoc, actually, and go back to working on G proteins. And I think I want to go back to my original career goal of running a research lab. But maybe I don't need to be so tied to what it is that I'm going to do with my career as far as research. And so I kind of decided to open up my mind to what research I could be doing.
For the first time since 9th grade.
Yes. Yes. For the first time since 9th grade. And I came to that realization, and I was really fortunate to be in Hugh's lab, because I went to Hugh and I said, "Listen, Hugh, I don't know if I can kill mice anymore as part of my job. And I've realized that this is just not the research that I'm passionate about. And I want to work on something that I'm passionate about." He tried to convince me with some projects. He's like, "Well, we could incorporate GPCRs somewhere here." And I was like, I still think I need to go somewhere else. I want to look more maybe at the molecular details, and maybe less on the kind of the overview physiology, but more molecular details and more get back to biochemistry as well. And so he was super supportive, though. He's like, I had his support looking for another position, and that was, I feel so fortunate. And that's another testament, kind of like the community and the department that I was in, I think.
Yes.
And it's interesting, you felt that the tens of nanometers, that gap of synapse that Hugh was studying in the nervous system was too big picture for you. You had to get to angstroms.
It's so interesting what big picture is to
Exactly.
biomedical science researchers. Okay, so you made it out to California for your second postdoc at Stanford, and you realized, okay, I want to do research I'm passionate about. That was another good starting point. When did you realize that you kind of found that? What was that particular experience?
I'll start at the beginning of my second postdoc, when I was interviewing actually for that role. And I had gone out and visited the lab, and at the beginning of the day I met with who would become my postdoctoral advisor, and he goes, we have the structure. And what he was talking about was the structure of the protein that I studied when I was a graduate student, RIC8, this one that folds G protein alpha subunits. Well, when I was a graduate student, people had tried to get the structure of this protein for 10 years or so. In fact, my first day as a rotation student in Greg's lab, I walked into his lab and saw these crystallization trays for trying to get the structure. And I remember as a naive rotation student looking at those trays, asking what they were, and he told me, and I said, oh, maybe someday I'll get the structure. And I remember him kind of laughing. He's like, we've been trying to get that for 10 years, and that's not going to happen. But I didn't know better. And so fast forward again to my postdoc interview. He's like, we have the structure. I'll have the postdoc show it to you, later in the day. And so I saw that structure, and I just started crying. And as I'm talking to the postdoctoral student, or trainee, he tells me where they got the protein. Well, I had sent protein when I was a student to Stanford to another lab, not even the lab I was interviewing in. And they had gotten the protein from that lab. And so it was actually the protein I had purified and sent. So I was looking at the structure of my protein, and I just started crying, And so then when I went back and talked to my postdoc advisor again, a year ago, he asked me at some point in the interview, like, you're already in a postdoc, why would you change? You could just continue on with your postdoc. And I just said, I want to work on something that I'm passionate about. I want to work on something that makes me cry. And then towards the end of my postdoc, that came kind of full circle as the project that I ended up working on, the time-resolved activation of G proteins through Cryo-EM. And we were actually able to get snapshots, progressive snapshots, essentially, of the motion of G proteins being activated. I was in a seminar listening to my advisor talk about this project and talk about the history of what led up to this project and seeing the whole story together. And I know where it's going. It's going towards my work as he's talking about all the historical things that have happened towards it. And I know where it's going, and I start tearing up. And then I start tearing up, and I realize I worked on something that made me cry. And then I just started kind of like bawling in the back of the room. And I was like, oh, my gosh, nobody look at me.
This is a real science nerd we're talking about.
Yeah, love it.
And so that's what I realized is working on something that you're passionate about. You can change the things that you're passionate about. But I think that's become really important to me.
So the research that made you cry led to you continuing that work. Eventually, you're here as an assistant professor. Can you talk about what your lab works on now?
Yeah, so that research that made me cry, the really profound thing about it was that a lot of the structural biology that had been done previously was on kind of like these static snapshots of these proteins. So it wasn't capturing really the dynamics of in our bodies, these proteins are actually moving all the time. They're not in these fixed, frozen structures. So this work actually was, for the first time, you could actually see progressive snapshots of what was happening. And so, if you think about what a movie is, actually, it's progressive snapshots. So you can actually now see these things in almost like a molecular movie of these proteins. And so I'm also still very passionate about G protein and GPCR research. So we continue working in that space, but we're studying, these at a molecular level in space and time, basically. Mm-hmm. So both molecularly, as we purify these proteins and then we can get their structure, and we could get these structures in more of a dynamic way than historically had been done. And that's really important because if you think about these GPCRs are drug targets, right? And so a drug target, you can kind of think of like as the target being a lock and the drug being a key. And so we use a particular drug that fits to that particular lock. And the thing, though, is that we don't always know what that lock looks like. And so we use kind of like a dirty key, which would be like a hairpin, and try and break it open and get it to work the way we want it to. But with structural biology, we can actually look inside that lock and see what should the key look like, and how can we develop a better key to initiate that activation of that, or inhibition, maybe we want to block that somehow, and use that for treating disease. And so we're now able to do that, and my lab studies that both at that molecular level and also in cells. Where are these G protein coupled receptors, and how do they interact with the proteins around them? So really studying GPCRs in both space and time, kind of in the cell. And so that's really the motivation of the research we're doing. And we also have an area of the research that's trying to develop new tools and instrumentation to be better able to answer these questions. And specifically, we use cryo-electron microscopy, which the university is, I think, investing in right now. And I was the first hire, I think, as a cryo-electron microscopy expert. And so I'm really excited to be part of that and helping other researchers here as well start to use cryo-electron microscopy in their own research.
So obviously your decision to do a second postdoc and to go to Stanford and to go to Dr. Skiniotis' lab to specifically learn how to utilize and take advantage of cryo-electron microscopy was a big factor in you now being our newest assistant professor in biochemistry and biophysics and pharm phys. Can you talk to us about how Cryo-EM is used as a tool to really study the temporal aspects of molecular interactions, which is what you've been talking about, sort of developing those motion pictures of molecules?
Yeah, so cryo-electron microscopy allows us to zoom in further than more traditional methods of microscopy. So light microscopy, we can get pretty high resolution things, but not as high resolution as when you use electron microscopy. So essentially with electron microscopy, you have an electron beam that's shot through at your sample. And so your sample is here, and your sample is a frozen sample in which either your purified protein or cells are frozen very rapidly. So it's non-crystalline ice. So unless your protein the electron hits your protein that's in this ice sheet, it can pass right through. So you can kind of think about it as if it does hit that, it's sent away and it's not going through. It's almost like a shadow on the detector. So what we're detecting, you can kind of imagine, is almost like our protein is like a shadow puppet in a way, like our hand, and it's in different orientations in the solution until you rapidly freeze it. So you have your protein frozen in this, very thin layer of ice. And that protein, because it was frozen very rapidly, but it was in solution before, you allow the natural dynamics of that protein to still occur. Unlike other methods of structural biology, such as crystallography, in which you've kind of locked that protein into a particular structure and you have a crystalline lattice of that protein in one particular state. In cryo-electron microscopy, or cryo-EM for short, you aren't limited to that. And so you have all of these kind of shadows in different orientations, you can imagine, or in different conformational states. And then when we process that data, we can actually tease apart those conformational states. And so you can utilize this tool that you have an ensemble mixture of proteins in different states instead of one specific state. And you can use that to look at, well, what are the proportions of these different states? Or something that we do is we can look at these ensemble mixtures at different time points of maybe an activation or binding to a drug or something like that.
Yeah. I mean, in a lot of ways, when I think about it, I'm picturing a crowd of people that are all observing the same thing, but maybe nothing's going on too much, and they're all doing different things. And you're able to visualize all the different things those different people are doing by their shadows. and all these different things, these different people are different molecules, all the same molecule but in different positions. And then all of a sudden something, the show starts and they all start to clap together. And that's like when you add your agonist or your antagonist, you add your drug that fits all those molecules and they transition. And now the whole population is kind of uniform, or mostly. And you can visualize that transition and the steps involved, and it's kind of it's a really neat process. So this is going to be a new technique, very expensive one, and require a lot of technical expertise that will be available to investigators here. Could you talk about your role in bringing Cryo-EM here and how you hope that evolves in terms of as being a research tool for the university?
So one of my hats that I wear is actually the scientific advisor to the Cryo-EM Center that we're building here. Part of that is, I would say, helping kind of give guidance on how to best set up that center based on my experience, but also being a cheerleader, I think, for this particular tool for scientists here. And so I talk to researchers all the time about how they can use Cryo-EM in their research, how it fits into the work they're already doing, actually. To expand that work. A lot of researchers have their favorite protein or their favorite topic and subject, and they've actually done all the hard part already. It's the biochemistry and the cell biology. That's really the hard part of this technique. The microscopy itself is not particularly challenging, but before you've been exposed to that technology, there's kind of like this intellectual barrier to getting into being able to do it. And so what I'm trying to do is, in my role, is I want people to know that cryo-electron microscopy it's not this unattainable tool that you need this, a lot of background in order to be able to do. It's now become a very accessible tool. And the university, I think, is investing in making this a resource. And so my job is to show people, lower that activation barrier of being able to get into this and use it in their research and show them, it's really not that difficult. You've done the hard part. Let me show you a few small things that you need to know in order to be able to do this. And as part of that, I'm working on getting some training mechanism workshops and things like that in place, as well as I meet individually with researchers often and talk to them about how they can use this tool in their research and give a little bit of guidance for them to be able to do that, what current research sources we have in the area to be able to use this tool as we expand. And I also am promoting collaboration between the universities as far as structural biology is concerned, and making University of Rochester part of that collaborative structural biology community in upstate New York. And as part of that, I actually co-organized a meeting with Monica Pillen at the University of Buffalo this past summer. And we actually had 150 researchers from across the state that were interested in structural biology, or structural biologists themselves, come and talk about their science. And I'm really motivated to be part of those kind of efforts and promote structural biology and cryo-EM in general.
And before we started recording, you and I were talking a little bit just, how's your week been going? And you kind of talked about the growing pains of starting a lab. I think it might be interesting to just touch on that a little bit, because now, you've had, I'm sure way back when, you had a vision of what running a lab was like, and now you're actually in it, and it does come with some things you have to do at the beginning to make it successful. How's all that been going?
In general, it's been going well. It comes with its little hiccups. I feel I was more prepared probably than most, because I think my graduate training environment, both by Greg and Bob, I was exposed to a lot of what faculty members need to be able to do and how to operate in kind of that system, once you become a faculty member. And so I feel fortunate in that regard, but then there's still things you can't anticipate. So one of the growing pains that we have now is every time I want to teach my students something, every once in a while it's like, I forgot to order that. This isn't an already set up lab. We don't have everything in stock. And basic things that I would have never had to think about, because you're starting from scratch. You need to build up everything in your lab and also build the people. And so my lab grew pretty rapidly once I joined in April. I think by June we had 3 graduate students, and my lab manager had joined. And then within the next month, I had 2 undergrads and a postdoc who also joined the lab. So I have 7 people. And so there's a little bit of growing pains in expanding kind of rapidly. But I'm so fortunate in the people that I have in my lab that I can't imagine my lab without any of them. And that's the part of the job that I think I enjoy the most, is working with the students. But the problem is I can't just work with the students 24/7. It's the other hats that I have to wear as well. I have meetings. It's fitting in working with students and being in the lab with the meetings I now have, or teaching obligations and things like that. So the juggling, I think that right now I'm in a period of becoming a professional juggler. I'm just in a learning phase.
And one of the things you get to juggle now that you didn't have to do so much of before is troubleshooting, I'm sure, which is a whole nother experience, trying to troubleshoot someone else's work. You know, it's hard enough to troubleshoot your own, but troubleshooting somebody else and you don't know exactly what they did to get to where the problem they're at.
That's true. I've now become a detective
There
as
you
well.
go.
I have small bits of information of why something didn't work.
Yeah.
Well, Makaia, this has been great. I think we'll get ready to wrap up here. Really appreciate you being on and talking about your journey. If there are trainees out there going through times of transition in graduate school, or folks, maybe some of our alumni who have now graduated and maybe are seeing a transition in their future, in their careers, what advice do you try to pass along now to folks like that?
I think that I would tell people that going through this transition period, I think at the time it seems very scary and chaotic often, because it's an unknown. We don't know the next step, essentially. I think in general things tend to work out, as you're going through these transition periods. But I think a lot of self-reflection, it would help them to have some self-reflection of what would make them happy in this transition. What is it they really want out of this transition? And also utilize the resources around them and realize they're not alone in feeling that way. I think that realizing that you have people around you have gone through this before, maybe not around you, but there are people who have gone through these transition periods before, and utilizing the people that are around you as you're going through these times of transition. You often recognize, I think, when you start thinking about that, you have a much bigger support and community than you think you have. When we feel like we want to feel alone, actually, I think we're not as alone as we want to make ourselves feel often.
All right. Anything else, Bob?
I just had one question. So now that you've come this far, you've spent a lot of time developing tools, reflecting on what interests you, what questions you want to ask. Have you considered, or do you have projects conceptualized where you might someday in the not too distant future actually go back to a problem that addresses maybe some of these molecular targets that have been more directly implicated through either genetics or biochemistry or pathology in Parkinson's disease, for example? Molecular interactions involving alpha-synuclein or Parkin or some of these other players. I mean, these are all things that, in theory, it seems like you could start developing projects for.
Yeah. Yes.
Or are you fixated on G protein coupled receptors for the long course?
I have learned not to be fixated on one thing. I think that my perspective has changed. Yeah. That I'm not fixated on one thing, and this is the thing that I always have to do. Mm-hmm. So now I tell people, I research the projects that I get excited about. And I get excited about a lot of different projects. And I'm not limiting myself to one thing. And there's the research projects, but now that I'm a faculty member and I have my own lab, there's also the people projects. I'm really invested in how my graduate students can be molded and kind of what their interests are as well. So I find that their interest is shaping what my interests are, because I like to see them excited about what they're working on.
And that's a really fun way to go, because you never know where it's going to take you.
Well, thank you again, Makaia. Any parting words for us? Anything we didn't talk about that you want to touch on?
I would just emphasize what a great community this university has. I think that that's the reason I came back, was the people, and the reason I was able to get through, I think, some of the harder times that I had. And the training that I received here was amazing. And so thank you so much for inviting me back and being able to tell my story.
Absolutely. Thank you so much.
Yeah, thank you. I really enjoyed being here today.
Thanks so much for tuning in to the latest episode of the Next Step podcast. If you liked what you heard, please share with other members of the University of Rochester community or anyone you think could benefit from these career stories. And we wish you nothing but the best in your own career journey. Until next time.